Hi, I'm Dave Robinson, and welcome to another one of my screencasts where I'll be using R to analyze data that I've never seen before. So as usual, the dataset is going to come from Tidy Tuesday, a project by the R for Data Science online learning community. And it looks like this week we're going to be analyzing pet names in Seattle. So this is exciting, all right. This looks like an open dataset of registered pets here, including license issue date, the animal's name, the species, breed, the zip code. Maybe we'll do some geographies and mapping with this data set. This comes from Jacqueline Norris, who's a friend of mine. And all right, looks like we're going to pull this data into R. Okay, so I'm going to create a new RMD. Library tidyverse. And create the Seattle Pets data. All right, looks pretty good. Oh, we're gonna just have to do a little bit of date parsing, and let's see what else we have. We have species. Okay, 52,000 pets. I'm gonna start just with a little bit of exploration by what kinds of species. So mostly dogs than cats, but a few goats and pigs. All right, that's good to know. 38 goats, 6 pigs. I really would have expected some rabbits or something. I would not have gone for goats and pigs as being the next two, but I guess that's why I don't live in Seattle. And I can also look at things like what are the most common animal names. A lot of these really look like, well, a lot of these look like human names to me. With maybe Luna is a bit of an odder human name, but it would be a strange name for humans. But I really have expected, I don't know, Fido. I don't know. When I was growing up, they had 2 dogs, one named Rascal, one named Lucky. So those are what I think of as animal names. But all right, let's see what else? What other columns can I look at? I can look at primary breed. Okay, and that's not pairs of breeds. It's like what types of retriever and golden. Okay, I'm looking at what do the commas mean. All right, I'm going to take a look at this, and I probably really want to clean the date at this point. So I'm going to remind myself I think there are a few ways to parse date. I wonder, library lubridate. I wonder whether I can just use MDY to parse this, whether we'll be smart enough to figure it out. I'm not so sure it will. This is month, day, year. Hey, look at that. It does. License, that's really great. It figures out, license issue date, and now I have it as a date. Oh, that's pretty handy. I can now look at license issue date, and let's say a histogram. See when most of these happen. Looks like a tiny number must have happened a long time ago, but mostly since 2016. 2016? I can try quickly count year equals year of license issue date. And see, yeah, almost all of them 2017, 2018. One that tells us we're not going to be looking at changes over time, because 2 years of data really, and then the bits of data before that, really is not enough time to look at changes over time. We may be able to look at changes over the course of a year if there's some trends there. I'd be skeptical if there was one. Okay, breeds. Yeah, so I looked at common names. I also took a look at, let's see, common breeds. That's probably one of the better graphs we can make, is this count primary breed. Oh, and we should certainly count the species while we do that. Species and primary name, primary breed. Okay, the most common cats. Let me start just with cats. So I'm going to start by saying I need to make a graph out of this. We've done this graph many, many times in these sessions. What I do is say I want top cat breeds, and I'll say primary breed and bar plot coord flip. I need to do a reorder there too. Actually, I'm going to do a couple things. I'm going to do put this after the I'm going to do a reorder. Primary breed by n. But do I really want n? I think I might really want percent is n over sum of n. Because it's not that meaningful to say, oh, there are 10,000 of these. I might really want to know what percentage of overall cats fit into this category. So I'm going to change some of these columns and throw in a scale y continuous labels equals percent format. Percentage of cats. 16 is too many here. So these are the most common cat breeds. Now that I've looked at cats, I probably want to look at both cats and dogs. I don't think I'm going to bother looking at species of pigs and goats, but that means I probably need to do a group by species, a top n 10 by percent. I could have done it by, and then, well, there's no overlap between dogs and cat species. This won't be an issue. I can facet by. It probably is. Let's see. Species. Let's find out what this looks like. I always forget to ungroup before I do this step. And I also need scales equals free. Both of them can be free. And you know, when I facet wrap like this, I probably want to stack the two graphs on top of each other. Yeah, that's a better graph for sure. Okay, this is pretty informative. So I can polish this up. One thing I'm going to do is set my theme. I also forgot this. Seattle pets. I'm going to do theme set, theme light. I'm also going to add a few labels here. I'm going to say primary breed, just write it a little better. Y is percentage of species. Sure. And it's a good start. All right, that's a pretty solid graph. If I want to make it a little more colorful, yeah, why not? What I do is fill species, but I would say I don't want a legend. That's neat. Okay. Oh, and I forgot about title. Most common cat and dog breeds. Breeds. I'd probably say of licensed pets in Seattle, 2017 to 2018. I say I could be a little more precise here. For example, if I threw in a month equals month of license issue date, I'd probably see, let me see, take that, view it. I'd probably see there was some month where it started being really common. Not really. Mostly just since 2017. All right, yeah. Technically, yeah. I'll say 2017, 2018. I could throw in a filter. Why not? What I'm going to do now is one last step. I'm going to throw in a filter to say license issue date is at least 2017 to make this accurate. And that way, if there were any other changes, this will still be, if there were any changes over time, this would be an accurate graph. Okay, so one thing I notice about this is cats are much more centered in the same few types. That actually tells me that I want free, wait, oh, I always get that wrong. It's a free Y even though it's coord flipped. Cats are really centered in domestic short hair. Like almost 1 out of 5 cats is domestic, in Seattle's report as domestic short hair. Whereas dogs are a little more spread out. You got your Labradors, your Golden Retrievers, and so on. All right. So that's a good quick visualization of dogs and cats. Now there's something I'm really interested in here. First, I'm going to remind myself. I'm going to remind myself. Let me see. If I count the species, cats and dogs, mostly dogs. Dogs also a little more spread out. I'm going to start just by focusing on dogs. And the question I want to ask is, are some animal names associated with particular breeds? So like more common in that breed than in other breeds. All right, to do that, or than they are in the overall population. To do that, what I would generally do is I'm interested in what are their overall rates of each name. So I can actually quickly say animal's name. They all have names, at least in the data set. And I might wonder, like, oh, is Lucy an unusually common name among Chihuahuas, for example? What I need to do is add a count of animals' names. I love that it has a plural there. I could... Yeah, why don't I quickly clean this up? Oh, oops, I forgot that this needs to happen in a cleaning step. Not that. My bad. This MDY has to happen in a cleaning step, otherwise that greater than isn't going to be meaningful. And maybe I'll rename animal name to animal's name. It's getting on my nerves, the extra S there. So I could add count, animal name. And you know what I could do is, so what add count does is add an N for each, I'm going to show that quickly. It shows like, oh, 2 dogs have the name Wally, 37 dogs have the name Mac. And Milo's a quite common name. So what I can do is add in a quick percent is N over, this is a little funny, over the number of rows. So what it says, this is the fraction of all dogs that have this name. So this is an overall rate. Let me see if I'm doing this right. Yeah, I'm going to say these are the overall rates of the dog. Sorry, pardon me, of the name. Yep. Now what I want to do is get it within each species. Okay, so what I can do, let me see, I'm going to do this slightly differently. Okay, I'm not going to do the overall percent. Okay, so that I'm going to do, here's my animal name and species. I'm actually not going to use group add count. I'm going to make this a little more explicit. Name total is N, and I'm going to then group, what I'm doing is now saying add my name total, and then I'm going to group by species, mutate, not species, primary breed, breed total is N, okay, and then I'm going to find the number of the animal name and species. So I'm actually going to, I can count by animal name and primary breed, and I'm also going to count by name total and breed total. So what this gets me is I can now get my percentages. So I can say, for example, so this now says the total for Mabel is, there are 27 dogs with the name Mabel. Total in this primary breed is only 1, and the total of let me go to the second one, Alfie. There are 19 Alfies. There are, I'm going to quickly reorder this to be, animal name, see, I'm going to throw in an ungroup here so the order is a bit more meaningful. Yeah, here it is. Here's how many, we have one Alaya, who's a Jack Russell, we have 2 of the Alayas, we have 400 Jack Russell Terriers, and 1 that is both, that is this combination. So one thing I know is that I'm not going to want anything that has too few of a particular name. So I can say, let me see, yeah, I can say name breed counts, and I can say filter name total is at least, I don't know, at least 50 dogs total, and breed total, only names and breeds that have at least 50 in them. Okay, so what I can then say is percent. So what percentage of Akitas are named Abby? Percent, sorry, I actually should have said what percent of this breed. Of breed, that's going to be n divided by breed total. And now I can say percent overall, which is in fact n divided by the number of items in dogs. So I'm going to quickly separate this. Here we go. So here's an example. There are percent overall. Probably really needed more like. Well, okay, so the challenge is that, it looks like, this is overrepresented because there's 1 and you would have expected there to be even fewer. But, of course, it's not very meaningful. I'm going to filter for some larger numbers first just to get a more general sense here. All right, and then then we can look at the ratio. We say ratio overrepresented. So how overrepresented is each dog? We can say is each name among the breed. So I can say percent of breed divided by percent overall. Percent of breed, percent overall. All right. There are 54 times as many Australian cattle dogs as we would have expected by chance. It's actually not really quite as impressive as it sounds because it's 7 out of 645. And we need to do actually a statistical test. I'm going to get in a minute to a statistical test we're going to apply. But at least this gives us a feel for what our ratios are. And now I can do overrepresented ratio, arrange descending overrepresented ratio. What is really overrepresented? We have loads of pit bulls, but we don't have loads. We have 2. And that's the problem, is we're always going to get these cases where... oh, shoot, I did this wrong. It's not n over n row of dogs. Wow, that's my bad. It's not n over n row of dogs. It's the name total over the total number of dogs. Oh, that makes a lot more sense. Okay, the story here is that I'd want to say there are 240. Not to look at the 7 of the total, but look at the 249 of the total. Sorry that this was a little confusing. I've done this before, but it's actually been a while, and I'm noticing how much time it takes just to do the bookkeeping here. I feel like there might be steps there might be ways to make this a little clearer. For example, we could have used TF-IDF, but I'm not going to do that in this. That's a bit of a different approach to this. All right, I'm going to stick to this. So here's what I was looking for. Beagles are overrepresented. Beagles tend to be named Lucy. What do I mean by that? I mean that we would have expected about 1% of dogs overall are named Lucy, but 3.5% of beagles are named Lucy. So it looks like it might be overrepresented. I'm going to turn down these thresholds a little bit. Yeah, so we would have expected very few Portuguese water dogs named Zoe, but we see 4. The challenge here again is that we're going to see random noise getting. I'm going to turn up the thresholds in a minute. We're going to see some random noise around here. And in fact, all of these might be due to random noise. What we want to work with here is really a hypergeometric distribution. I'm going to use, let me see. Yeah, let's use hypergeometric. Okay, so we want to know, are Lucys overrepresented among beagles? Now, we know that across the entire set of dogs, there are 337 Lucys. And we know that across the 542, there are 19 Lucys. So our alternative hypothesis is that there are... our hypothesis is that null hypothesis is that you have the beagle has the same chance of being Lucy as an overall dog does. So that would mean percent overall being here, that the true probability was 1%. Let's discuss the hypergeometric distribution. This is popular in some slightly old school ways of looking at what we call gene enrichment, back in my old field of bioinformatics, where we would ask, are there more genes in this group than we would have expected by chance? More genes that are, say, change in an experiment that are in this particular gene set. So what we're looking for is like dog name enrichment. So I'm going to ask here is P is, let's see, yeah. What we're imagining here is the number of white balls drawn without replacement from an urn that contains both white and black and white. And the overall set is going to be the total number in the urn. You know, we could have used the binomial here. It's so much easier. I'm going to use the hypergeometric. It's showing off a little bit because it's more statistical rigor than we need. But here is the story. We want to say, I'm doing a one-sided t-test. One-sided hypergeometric test. I'm going to say p hyper, where the hypergeom p-value, where we say, first, here's the number n of beagles named Lucy. 19 beagles named Lucy. And what are the overall white and black balls? That is the overall number of Lucy's. It's 337. Name total. What's the total number of black balls? That is dogs not named Lucy's. At least Lucy. I'm going to quickly pull this out. So now we say number named Lucy, number overall minus the ones named Lucy. So I'm computing the P, meaning the cumulative distribution, and the number of balls drawn from the urn is the number of in this breed. So 500. We had a large urn with how many is this? 30-something thousand. We had an urn with 35,000 balls in it. 337 of them were white. That is, were named Lucy. We drew 542 balls out of the urn, and we saw that 19 were white. And we're wondering, did that happen by chance? So each of these. Okay, this I think is the one-sided hypergeometric p-value. Now I can yes, total. Cool. It's actually 1 minus because probability less than. All right, so one thing we can say is beagles are absolutely, well, not absolutely. I haven't adjusted for multiple hypothesis testing yet. But we can say that's actually a really strong p-value. Why are we seeing 19 out of 330? The fact that we see 19 out of 542 beagles named Lucy, that is overrepresented. It's unlikely we would have gotten 19 by chance. And now that we have this, I'm going to say I'm going to call this hypergeom test. So it's the hypergeometric distribution. I'm going to show, I didn't really introduce it from scratch. This isn't the right setting to, I don't have a lesson plan or anything. I just said I'd show it. Hypergeometric distribution is a probability distribution of probability of k successes in n draws without replacement from a finite population. And that's what we're seeing here, is this finite population of dogs. And if we assumed that beagles were drawn randomly from them without being more likely to be named Lucy than anyone else, that's where we would be. That would be our null hypothesis. So here's our hypergeom test. Whenever you have a p-value distribution, p-valued histogram, you have to make a histogram out of it. This is one of my older blog posts where I read about how to interpret a p-value histogram and show some of the things you learned from it. So I'm going to show this p-value histogram, and we'll do some interpretations. Remember that it is a one-sided p-value, which means that if, let's say, a dog name is underrepresented, it's going to be show up. It's going to show up as a very high p-value. A complication just hit me that we're going to have to take a look at. So we see, in general, our p-values, I'm going to make the bin width a little wider. In general, our p-values are shifted towards lower values. This is a promising sign. These are what we call an anti-conservative p-value shift. What it suggests is that some breeds actually are underrepresented. I say that, but we're missing something really critical here, which is we don't have any zeros in this data set. The zeros got left out. And this makes me realize I need to refactor how I did this approach a little bit. What I can do is say, let's make these summaries. And this is name counts, filter, name, total. I'm still looking only at the big groups, and I'll get into later into why that's important. So I'm looking at name counts greater than 200. I'm also going to take a look at same thing by breed, by primary breed, breed total. Oop. Now I can skip this. Now what I can say is here's my breed counts, here's my name counts. Oops. 200 is way too high a threshold. Oof. Now I have to pick these kind of accidentally, which is kind of arbitrarily, which makes a risk of p hacking. I wonder how much 100 would be. Let's start there. Okay. And what I would do then is look at our dogs, count by primary breed and animal name. And then, let's see, remind myself I need to, yes. What I do is complete. What I can do is say complete primary breed and animal name. Let me quickly check. I suddenly worried this is going to crash something, so I like to run it on a smaller subsample first. That was fine. Yeah, this is fine. I'm going to go ahead and run it. Yeah, it ends up being quite a large table, but this is like primary breed and animal name. Now I'm actually, once I've got those, so now we actually have zeros built in. So the fact there are no Abruzzese Mastiffs named Aaron is now a piece of information. This got really big. We could have reordered a little to avoid that. I'm not going to bother because I'm going to do a right join at this point. I'm sorry, an inner join with our name counts by animal name and with our breed counts. And I no longer need these two. This is still useful. I think the rest of it is basically still useful. I'm going to start with this step. Notice it's now smaller. I did my filters already. Okay. And what I want to show is this. There are loads of Labradors named Lucy. If I scroll all the way down, I should... Yeah, here are all my zeros. So there are zeros in this table now. It's only among this particular set of animal name, of 37 animal names, and how many primary breeds? 40 primary breeds. But in this combination, we have all we include the zeros. So here are our breed counts. P-value. I'm going to show other ways to compute this, but I'm going to start here. Here's our hypergeometric p-value. All right, and hypergeom test. We now have recombination including our zeros. It didn't change it a lot. It's actually kind of this shape is super weird. what this is suggesting in a one-sided p-value is that there are no dog names that are there are basically no dog names that are underrepresented. We see ones that are overrepresented, but there is a paucity of ones that are underrepresented. I can think of a few reasons that would pop up. I'm not going to dig too, maybe in a bit we'll dig more into the p-values. I did a lot of my research in graduate school was on looking at distributions of p-values under conditions a little bit like this. Maybe we'll dig in a little bit more. I think it has to do with the fact that we set thresholds, particularly common names, is that the kinds of names that are underrepresented in multiple common breeds would not be showing up in this data. But actually, now that I'm thinking about that is kind of an issue with the data. It's probably not a big deal if what I go ahead and do is I say, let's look at and this is a problem. I'm going to keep an eye on this. This peak around 0.5, it may or may not be meaningful. I'm going to make this a little wider bars. It may not be meaningful. I think mostly it's just a decay here. All right. What I'm going to do is start with a conservative approach. I'm going to say, I really can't, can I? Okay, no, all right, yeah. What I do is these look like they're significant changes. Unusual number of Havanese named Oscar, of boxers named Rocky. All right, that makes, of course, we would get your boxers named Rocky, just like the movie with the Rocky Balboa. And it looks like pit bulls with the name Rocky. Each of these, maybe they're overrepresented. Maybe not, though. We are looking at 1,480 hypotheses. So we can't just look at these p-values. We're going to have the problem of multiple hypothesis testing. I'm going to add a step here. We have a few ways of correcting p-values. One is called Bonferroni. Actually, take it back now that I remember. It's not quite, the default isn't quite Bonferroni. 1 second, I remember. It's, I'm going to start with, just quickly looking through a second. Dominated by Holmes method, that's right. I'm going to start by using Holmes method. So this is is that the default? Yes, it is. Okay. So so what I'm going to say is Holm p-value. This is me correcting basically it's trying to avoid having any false positives. The same in the entire set of p-values of hypotheses that we're willing to reject the null. So I'm going to say is p.adjust hypergeom p-value. Here's my adjusted p-values. Make this a little wider. All right. What this shows is that some of these, like Havanese named Oscar, having 8 out of 466, it does look like it's more than we would have expected by chance. Even correcting for all of our hypotheses, we're seeing this and boxers named Rocky. Those seem to be significant. If I filter for Holm p-value under 0.05, pretty typical setup, what I see is we see again Beagles named Lucy, boxers named Rocky, and Havanese named Oscar are a little bit overrepresented. Start. However, this is not the only way to correct p-values. There's a slightly more common approach when you're working with not just, say, dozens of hypotheses, but thousands. Notice how much that changed the p-value. We went from an insanely conservative p-value, low p-value, to this fairly high one. And the reason is when you have this many hypotheses, these methods have a really sharp effect on your power and your ability to detect changes. There's another approach called false discovery rate control. Probably the most notable implementation of it is called, we have a description here, Benjamini-Hochberg. So it's BH, or I can say FDR for false discovery rate. So I'm going to call this FDR is p.adjust hypergeom p-value, and say method equals FDR, false discovery rate control. So at false discovery rate control, the difference between that and a corrected what they call family-wise error rate that Holm uses, is that this family-wise error rate is trying to say what is the probability, trying to ensure that you don't get anything wrong. That is, of these three that we accepted. Of these three that we rejected the null hypothesis, trying to ensure that that less than 5% of the time, if they're all null, if any of them are null, that they would be rejected. That's actually a really high standard to hit. We're often interested in, we want to keep the fraction of these that are incorrectly included to a low reasonable level. And it's a really subtle difference, and sometimes it involves a little bit of investigation. It involves like reading some papers, having a better understanding of it. I'm not explaining it all that well myself. Screencasts aren't the best way to do statistical education. But what it means is I can do a filter for it, and what I notice is it has a higher power. I have the ability to, I can include a couple more hypotheses, and what false discovery rate is saying is it thinks that in the long term, less than 5% of these, or about 5% of these, we expect to be incorrect conclusions. So there are only 6 of these. So we would say, on average, all of these are overrepresented. So this was a use of multiple hypothesis testing, where we were able to get a little more power by using the FDR. Here we go. Yeah. We were doing it now. We were doing control of FDR, where we're saying we're able to include all of these, and our expected false discovery rate among them is still below 5%. That's like I said, a more common approach. So here are 6 conclusions. We think that Oscars, the name Oscar is overrepresented among Havanese. We think that Milo is overrepresented among American Cocker Spaniels. We tend to see pugs named Zoe, and beagles named Lucy, and Labrador retrievers named Scout. That's pretty fun. That's a fun thing that we were able to learn. Let me see. Yes. So that was us. We did some hypothesis testing on these names. Could I make a graph? I'm not going to make a graph. This is probably best done as a table. So I would probably start by saying, excuse me. I'd probably at some point show this visualization, and I would note, as I was saying before, this is a reason to be concerned. Where are our underrepresented pairs of animals and names? I'm thinking for a second as to how. If a name never appeared among the animal or the primary breed, that would be one reason. I'm looking at common names, looking at common breed counts. Yeah, it's not just none of them are significantly underrepresented. It's like we're specifically missing ones that are underrepresented. This is where a little bit of statistical critical thinking comes in, and it's hard to do under pressure. Let's try and answer this question about names and animals being associated. Okay, so what we could start by saying is, how many did I filter this down to 100? What did look underrepresented? Probably the zeros. If I look here at my hypergeome tests and I arrange by, let's say, descending hypergeome p-value, it's all the cases, well, not all entirely cases, generally cases where n was 0. Did I look at, were we underrepresented in zeros in this data? Like there weren't a lot of cases where an animal didn't have enough, or is it something different? I think I have a suspicion. All right. Here's why we're not quite seeing a well-behaved distribution. There it is. All right. Why aren't we seeing a well-behaved distribution? It's because your number of animals with a particular breed and name cannot go below 0. So if I look at these, there are 315 combinations here where there was just no animals with that name. So if I look at their p-values, if I histogram case with zeros, some of them that have 0 have hypergeometric p-values all the way at 0.4. So that's actually nuts. What that's saying, I see what it's saying. What it's saying is that these would be the ones. Oh, where's my breed total percent overall? It's the ones with the If I look at this and here's all the zeros. I'm going to arrange by the hypergeometric p-value. And we see is it's the breeds that are only around 200. We're going to see a really wacky distribution. This is because we're in particular we're going to see that because we see that the p-value is stuck around the middle. And that means these p-values can never go below 0.44, 0.45. That's why we see a drop off at the high end of the spectrum, is this lower bound on a p-value. Okay, it took me long enough to get there, but since that's the main reason. If I'm scrolling up, I actually would have expected this to look a little different. I would have expected. Oh no, I guess it's actually not crazy. Basically, as your here we go. As your number of dogs with this name goes up, or your breed total goes up, or no, in particular if your breed total goes up. Like here we have our ones where, like a Chihuahua short coat, where there are thousands of them, and not a single Oscar in the bunch. And that has a hypergeometric p-value very close to 1. But it's impossible for your smallest smallest breeds. Okay, so that's something we learned, and that's why we're seeing distribution. How can we dig a bit deeper into this? The way I would think of it would be, I would say, let me see. I would say I would look at what the maximum one-sided p-value would be at different number of breeds. So if I said, could make a graph of this. Is that what I should do? Should I make a graph of power? Okay, I'm going to try it. No, I'm going to start with this. What I'm going to say is, imagine we had a hypothetical dog with 100. Is that the minimum? Let's do 200. And what if breed total went from actually don't this I can do data frame. What if breed total actually no wait. I'm using crossing because I want every combination of what if there were 100, 200, or 300 dogs with this name, and then what if our breed total went from 200 to 1,000 by 25. And let's say n was always 0. The number of dogs in this breed was always 0. The total dogs we also keep to be constant. Then I would say that hypergeometric, the one-sided p-value here it goes. Okay, and I would do a visualization where I would say, as the size of your breed total increases, color equals factor of name total. I'm going to call this min p-value, because this is really, what would it be if it was 0, if n was 0? Be really clear what we're visualizing here. Yep, here it is. Okay, so this is if I need to communicate to someone. Someone asked me, why do you have trouble? Why would you have trouble detecting? Particularly, why would you have trouble detecting underrepresented dogs? What I would say is, here we go. X is total number of dogs in breed. Y is minimum one-sided p-value, number of dogs with name, And this gets the idea across. You can't, if you don't have a lot of dogs with this name, and especially if you don't have a lot of dogs in the breed, yeah, this is your, oh, I said minimum, it's maximum. Max p-value. And that's why we see a break here. Another way we could have said this would have been in terms of statistical power to detect changes. That is 100% a problem that we have. This is kind of fits with this. If you can't see your things going the other direction. Having said that, it's not a big concern for the method that we're using, that the histogram is not it drops off close to 1, because while we can't detect changes in the 0 direction, that doesn't mean we can, because of the you can't have fewer than 0 names. It doesn't mean that you can't detect changes here in this region unless you're using a very particular set of statistical tests. Then why did I go through this process of figuring out what the maximum p-value could be? The reason is this section where I point out that if you see some funny shapes, like if you here in this blog post, how to interpret a p-value histogram, I say if you see some really weird shapes, like this, like here is dropping off, something's strange, something doesn't look like anything of the post, stop whatever you're doing and find a statistician. There may be a simple explanation, but you want to make sure you found it. So I want to take my own advice and say, when I look at this histogram, do I understand why we have a drop-off close to 1 on one-sided p-value distributions? Let me make this super clear. X is one-sided hypergeometric p-values for overrepresented name. Okay. All right. So now that I figured that out and I feel comfortable that I can apply the false discovery rate test, that's a little hard to explain why I feel comfortable with that. Like I said, I just have to spend some time working on with p-values and understand the math behind it. We're able to report each of these. What I would probably say is take this data, select primary breed, animal name, and I might rename it while I'm at it. This is the kind of thing we do if we're breed, name, dogs with name, and now I'd say percentage breed of total, percent overrepresent, remember this now, percent overall, percent of breed, percentage of breed, percentage overall. I'm putting this in a table, so I probably want to throw a little bit of library scales. Where did I do this wrong? Percent of breed. Oh, it's not select anymore, it's transmute because I'm changing the columns. All right. I also neglected to do my filter. Oh, and p-value uncorrected. Do you even want to include that? Probably don't want to report it. Don't want to get people excited. There are a few ways one could communicate this. I'm going to start with FDR adjusted p-value. Filter FDR less than 0.05. And I would turn it into a little bit of a table, and this would look all right in whatever I was visualizing. I probably want to do a range to ascend FDR. So what I find then is we're pretty confident that Lucy's named Beagle. That Beagles are more likely to be named Lucy. Zoe and Pugs. Well, yeah, it's still the adjusted p-value we still see significant, but if you want to have a little doubt about any of these, that's the one. we expected 0.3%. It looks like there were actually 1.44%. Okay. Oh, and I should probably note one-sided p-value. So what do I mean by one-sided p-value? What I mean is that we are not allowing here the hypothesis of names being underrepresented, and that means it's a somewhat controversial approach to use a one-sided p-value because it doubles the region where you can the p-values would be twice what they were here if we were looking at a two-sided. Sorry, the p-values are half what they would be if we were looking at a two-sided p-value. Actually, I can't really go into the full depth of the controversy. Like I said, I haven't prepared to teach and discuss this, but I'm sticking with it just because we know we're looking only at overrepresented names, and in fact, as we saw, we wouldn't be able to find many underrepresented names even if we were trying, because you can't go below zero. Your frequency of names can't go below 0. So I felt somewhat comfortable in this case. Okay, and this would be the if I did set a p-value threshold, I'd probably just remove Zoe the pug, but I would see these other cases. So, my favorite conclusion is that boxers are unusually likely to be named Rocky. I think that's pretty cool. I'm also curious about Lucy the Beagle. Is that a thing? Is there an Lucy the Beagle? Is it a favorite? Is it a famous beagle? I don't think so. Lucy the counter surfing. Oh, here it is. Lucy the Beagle is like a series of books, I think. Lucy the Beagle book. Oh, absolutely Lucy. Oh, okay. So that looks like that's one explanation, I think. Lucy is a children's book where Lucy is a beagle. Oscar Havanese. Do we have an explanation of that? Oh, sorry, it's super cute, but I don't have Oscar the Havanese. This is not a very popular dog, so I don't know that would explain the reason. So I don't have a good reason here. Milo Cocker Spaniel. How many dogs was that? Only 7. I don't want to. Yeah. I don't have an explanation, but like I said, how about Scout Labrador Retriever? Yeah, I'm not seeing much here. It's definitely a good name for a dog, Scout. I wonder if there's anything Seattle specific. Nah, I'm not seeing much. Oh, so there's a yellow Labrador Retriever, Scout. All right, so I'm not going to dig too deep. I don't want to read too much into this. And this is only a handful of dogs. But I do like that that Lucy, this is this overrepresented result that really kind of does stand up to a little bit of statistical scrutiny, and and we found a couple of dogs that were overrepresented. So this was a really fun screencast for me. It was a little different than my usual approach. We made a graph. We made this this graph of common. Oops, I'm gonna remove this. We made this graph of common cat and dog breeds. But we also looked at were particular dogs more likely to be were particular breeds more likely to have particular names than others. We did some hypothesis testing. We did a little bit of statistical detective work where we said, why does this distribution not it should look uniform for higher p-values, and we found the reason that it doesn't. And we did we came out with some conclusions and some statistical caveats to them. So that was like I said, it was a little bit of a different examination of what dogs are more likely to have, what names. And one of the reasons we had to do this kind of statistical examination is that our numbers aren't that high. If we had 100 million dogs, we probably we wouldn't need to do so much examination. It would be very obvious that this dog breed, this name was very overrepresented in this dog. But if we say, oh, we expected 1% of dogs to be named Lucy, but in fact it's up to 3.5% of beagles, and that's 19 out of the number of beagles, we're less sure that we can say, oh, that's definitely the overrepresented. So that gives us a chance to really break out our statistical testing a little bit. Okay, well, thank you very much for joining. I hope you had some fun doing some statistical analysis with me, and I'll see you next week.